My name is Karthik, and I'm part of the R&D team at Biocon. And so I don't have any association with Indulge. So I have been in Biocon since 2005, fresh out of college, and I've been ever in Biocon since then. So what's our insulin glargine journey very briefly? So I lived through that avatars of insulin glargine through almost 15 years, starting from 2006 until 2021. The reason I'm talking about is 2006 is when we started. 2021 is when we got the interchangeability status in the US. So in that 15 years, what happened? Biocon's philosophy of product development changed. Regulatory landscape was changing. Our aspirations were changing. So everything was changing all the time in these 15 years. But we started with India-centric focus. Then we made it as we wanted to get into other important emerging markets. Then we said we also wanted to crack the EU and the US. And so we started with probably no guidelines in 2006. Then I think 𝐬𝐸𝐚 took the lead in providing certain product-specific guidelines, and insulins was one of them. And FDA did not have any guidelines at that time. So it was pretty, I would say, it was unclear in terms of their expectations. And then we had also this partnership that I think now it is called Viatris. Mylan came on board. So we had to kind of agree on the product development philosophy. And then obviously we had to engage with the agency multiple times. It was new for all of us. And we are doing insulin glargine at massive scales. So we are talking about not 2,000 and 3,000 liters. We are talking about 30,000, 50,000, 60,000 liters of fermentation. And I think what this journey taught me is insulin or insulin analog can be very small in stature, but it can give you nightmares. And I think all of the people, Lakshmi has worked on this, Naren has been part of this journey. I think my colleague, Dr. Gurusharan, has been there. Sethu is here. Okay, so he's the manufacturing head for us. And so all of them can vouch for it, that doing something at that scale, even if it is 51 to 52 amino acids, can give you nightmares. Okay? So a small, so-called small molecule can really, really humble you multiple times, and which is what happened in these 15 years of work. Then I think few things, anecdotal events that happened is, so we started it was not a monograph product, so we had our own methods. And as luck would have had it, when we were into the clinical trial, monograph was published, and as expected, it was a different method that was being listed. So we had to take into account the differences that you see between the product quality of both yours and the reference product with your own methods. So we had to account for that. Then we said, we started the clinical trials, and then we had a flash of brilliance that we will take it to a different site because for commercial reasons. So we started the clinical trial in one site, and then we said we want to commercialize from the other site. So that was one, I wouldn't say it is a mistake, it was a business need, but it does come with a lot of baggage, especially when you run a clinical trial from one site and you want to commercialize from a different site. Then the product quality itself. So we had a different expression system to make things interesting. It was not in E. coli, it is in Pichia pasteuris. Now, the way 𝐬𝐸𝐚 and FDA were perceiving the risk in terms of the product quality was different between these two agencies. So FDA was okay. They said, you're running a clinical trial, we don't have a problem. So there should not be risk in the product quality, even if you have minor glycoforms. Now, 𝐬𝐸𝐚 said that, no, we don't agree to that. So you have to control the glycoform. But you can do the clinical trial, but when you commercialize it, we don't want to see it. So like this, there were a lot of iterations that happened. So it was very difficult to essentially meet the expectations of both the agencies and keep them happy. Let me put it that way. So, and then subsequently, so we did that clinical trial with a certain variant in the product. We did commercialize it with removing that variant. Although we proved that it was low risk, there was a perceived risk in the minds of the regulator, especially the EU regulators. And so we had to do multiple trials. In fact, the joke within our organization is that we probably did more bridging clinical trials than even the innovators. So that was the number of trials we had to do in order to convince the regulatory agencies so that there's a single product quality that goes to all the regions. And that was a humongous task for all of us. And during all of these stages, okay, management will continue to take their decisions, keeping in mind a certain focus. So all the technical teams, including manufacturing, including quality, including R&D, all of us had to ensure that we continue to bridge the product quality as we were moving along. And so trust me, it is about 15 years of worth of data, right? And somebody in the audience today asked, what will you do if you are having historical data? Do you still retain it? Because it evolved. And our quality management system was also evolving during this process. Meaning from, I would say, less oversight from an R&D perspective, we literally, I think the R&D quality management system actually came on par with typically what happens in QC today. That is the level of data questioning we do today so that we will never ever have to worry about data integrity. And I can tell you that in all of these first line of products that we have gotten approval, we were audited by FDA. R&D labs were audited exclusively by FDA for 2 to 3 days. And they never found any data integrity issues. They were so happy, and they felt that probably we are overdoing a little bit, but it was still something that they were comfortable with the quality of data that went into the package. And the most important thing in terms of the regulatory agency approval was that until March 23, 2020, insulin was being regulated as a new drug molecule, okay, via the NDA route. Okay, it was not treated as a biologic. I don't know how many of you knew about this, but insulin, the human insulin and analogs were approved as a small molecule. It transitioned into the biologic route as we talk about the 351K, the BLA, in March 23, 2020. So we were the last organization to file via the 505(b)(2) route and get approval via the 505(b)(2) route. After we got the approval via 505(b)(2), we had to transition to 351K. So we went through another round of regulatory approval for a year, okay, before we got essentially approval as a biosimilar first via the 351K. And subsequently, by virtue of declaring the product as a biosimilar, we got the designation as an interchangeable biosimilar for the first time, okay, for any company in the world, because there was an insulin-specific guideline that came out in US FDA, by US FDA in 2019, that insulins can be considered as interchangeable, okay, because interchangeable is a concept that exists only in the US, not anywhere else. Interchangeability is a concept, as on today, a legal concept in the US where the pharmacist can substitute if a prescription is written without consulting the doctor who writes the prescription. So as you can see, over a period of 15 years, from being India-centric to few important emerging markets, then to US and Europe, and then trying to strike a balance between US and EU needs, and transitioning to essentially interchangeable biosimilar. It was like a Herculean task for all of us over the years, but worth it when it happened finally. But I think as Jananjay said, I think by the time we got to that, I think we were like, okay, fine, it happened. So thank God. So we just lost that. I don't think we lost the mojo of basically doing things, but because the regulations had changed so much and the perceptions had changed so much, it was a kind of a big relief rather than I would say, I don't think we were ecstatic, but we were actually saying, okay, I just got done with the leave me. So with that, I'll just stop here.